好，嚟到2017年嘅第10條題目呢，就係基礎遺傳學啦。今次我哋嘅切入點呢，就係一個人捲到脷，抑或捲唔到脷呢個能力啦。咁啊，1940年呢，咁有個科學家就提出一個假說啦，就係講啦，捲唔捲到脷嘅呢個能力呢，係由一個單一嘅基因所去控制嘅。咁所以啦，佢就收集咗唔同類型嘅數據啦，咁啊睇下啦，父母親當中佢哋都係捲到脷嘅話，究竟生出嚟嘅小朋友捲唔捲到脷呢？個數字會係幾多呢？另一款嘅個案呢，就係父母是但一方呢，就係捲唔到脷嘅，咁生出嚟嘅小朋友啦，究竟又有幾多捲到脷，同埋有幾多係捲唔到脷呢？咁第一部分呢，就問啲直接啲嘅分類題目啦，佢就問翻我哋呢，捲脷嘅呢個能力，究竟佢係連續變異啊，定係不連續嘅變異呢？跟住要解釋我哋嘅答案啦。咁今次呢條題目啦，自不然就考下我哋對於連續變異同埋不連續變異嘅理解啦。咁而家實質啦，對於捲脷呢個能力呢，我哋係得兩類型嘅表現型嘅啫，就係捲到脷同埋捲唔到脷。佢哋之間呢，係冇一個中間嘅特性嘅。我捲到少少咯，我捲唔到少少咯，係冇嘅。你一係捲到，一係捲唔到。所以答案咧都唔難理解啦，就係不連續嘅變異啦。咁啊，因為啦一個好明顯嘅分類就係捲到脷同埋捲唔到脷，而兼且係冇一個 intermediate 嘅 category 嘅。跟住啦，去到第二部分咧，咁呢位科學家咧就嘗試跟翻當初阿曼德爾嘅概念啦。咦，有啲表現型咧係顯性，有啲表現型咧就係隱性嘅。咁當佢收集咗唔同嘅數據之後啦，佢就得出一個結論啦，就係捲到脷咧就係一個顯性嘅表現型，而捲唔到脷咧就係一個隱性嘅表現型。咁題目就問我哋啦，根據翻數據啦，就解釋下點解呢個科學家會得出呢個結論啦。其實呢條題目咧就係考翻我哋無中生有同埋四步成詩啦。咁無中生有咧，我哋就要揀翻邊一啲嘅個案係可以幫我哋去得出呢個結論嘅。咁自不然啦，無中生有就係捲脷生到個捲唔到脷啊嘛，呢啲就叫無中生有啊嘛，係咪？咁所以我哋抽取嘅咧一定係 case one 噶啦。咁但係點樣去表達我哋嘅答案呢？就係四步成詩咯。成個概念啦，我哋要諗下啦，父母親嘅表現型啦，去推斷佢哋嘅基因型啦。跟住啦，又諗下細路哥嘅表現型，就又去推論下佢哋嘅基因型啦。而去到第三部分呢，就要喺呢個個案入面咧去揾翻邊一啲嘅個體呢，就係集合型。咁今次呢個情況呢，就係父母親係集合型嚟嘅。第四步呢，就能夠判斷得到呢一個顯性嘅特徵呢，就係捲到脷。而隱性嘅特徵咧就係捲唔到脷啦。我哋睇下點樣表達我哋嘅答案咯。第一啦，揀翻 case one 先啦，無中生有揀出嚟噶嘛，係咪？跟住啦，睇下父母親又好，或者睇下仔女都好。我哋睇翻仔女咧就最容易理解噶啦。對於呢個捲唔到脷嘅後代，佢都有一個捲到脷嘅父母親，亦即係話呢個捲唔到脷嘅後代，佢會喺佢捲到脷嘅父母親咧去接收到起碼一個係捲唔到脷嘅等位基因。咁所以就會知得到啦。即係話父母親咧就係一個集合型啦，佢表現出嚟嘅就係捲到脷嘅呢個特性，但係佢自己內在咧就有一個捲唔到脷嘅等位基因，即係意味住啦，嗰個捲唔到脷嘅等位基因咧係被遮蓋住嘅，係表現唔到出嚟嘅。咁所以我哋就知得到捲唔到脷就係隱性嘅表現型，而捲到脷就係一個顯性嘅表現型啦。但係呢條題目點解得2分呢？過往嘅四步成詩咧係4分噶嘛，係咪？咁啊，因為啦，今次第3、第4步咧，其實題目已經講咗俾你聽噶啦，尤其是第4步。而第3步咧，你就算唔講佢係咪一個集合型嘅基因咧，你都會知道啦。由於父母親就係擁有捲到脷嘅等位基因同埋捲唔到脷嘅等位基因，而由於佢哋自己係捲到脷，即係話啦，捲唔到脷嘅等位基因咧係被遮蓋咗，表現唔到出嚟嘅。好，咁頭先 part A 咧，我哋就透過收集唔同類型嘅數據啦，就去研究下啦，究竟捲到脷嘅呢個能力咧係咪受單一嘅基因咧所去決定嘅？咁啊，去到 part B 咧，我哋有另一款嘅探究咯。1965年咧，我哋就去揾一批捲唔到脷嘅父母親，就係睇一睇究竟佢哋生出嚟嘅小朋友咧，究竟識唔識捲脷咯？咁後尾我哋發現啦，超過30%嘅後代咧都係捲到脷嘅。佢就問翻啦，呢個結果能唔能夠支持到喺頭先 Part A2 嘅結論啦？跟住解釋下我哋嘅答案咯。咁啊，成條題目啦，嗰個思考邏輯咧，我哋就要去理解翻啦。頭先喺 Part A2 我哋知道咗啲乜嘢？就係咧，捲唔到脷係隱性嘅表現型，而捲到脷咧就係一個顯性嘅表現型嚟嘅。就當我哋啦，叻翻啲大世界啦。 即係大階 T 咧就係捲到你嘅等位基因，而細階 T 咧就係捲唔到你嘅等位基因。話啦，一啲捲唔到你嘅父母親咧，佢哋嘅基因型咧一定係細階 T， 亦即係話喺我哋 Part B 去揾嘅呢一批捲唔到你嘅父母親咧，佢哋基因型應該就係純合嘅隱性啦。咁究竟得出嚟嘅結果竟然有捲到你嘅後代，合唔合理呢？其實係唔合理嘅。咁啊，因為啦，如果你夠快手去畫呢個基因圖嘅話咧，其實10秒畫咗噶啦，你會發現啦，生出嚟嘅全部後代都應該係捲唔到你先真嘅。但係無啦啦點解會有啲捲到你嘅情況出現呢？咁所以啦，就係佢不能夠支持個結論。因為如果當呢個捲唔到你嘅父母親，佢哋係純合隱性嘅基因型嘅話咧，邊有可能生到一啲捲到你有個大階 T 嘅後代呢？係咪？咁其實呢一條題目呢，都係反映咗少少嘅科學本質㗎。科學嘅知識係暫時性而可變更。你諗下，冇錯喺 Part A 呢，佢同你講啊嘛，捲到你呢就係顯性，捲唔到你呢就係隱性。但係去到 Part B， 我哋發現好似唔係咁啱，有啲怪。咁究竟發生咩事呢？科學係一個不斷發問嘅過程啦，而當中啦，懷疑論亦都可以促使呢個科學嘅進步嘅。因為其實你睇翻 Part A 呢，個科學家佢提出嘅假說都係想去驗證一下呢，究竟捲唔捲到你嘅呢個能力係咪受到單一嘅基因所去決定嘅？其實到頭來佢係冇肯定得到佢係咪受單一基因所控制嘅。佢純粹就係話俾你聽啦，嗱，捲到你咧就係顯性咯，捲唔到你咧就係隱性咯。咁你信我啦，連自己最基本嘅問題都答唔到，我好難信你嘅。OK， 好，咁經過咗 Part A 同埋 Part B 嘅研究啦，咁我哋對於捲唔捲到你呢個能力啊，呢個特徵咧嘅研究都仲未得夠清楚啊。所以啦，我哋未死心嘅，繼續探究落去。1971年咧，我哋就揾咗一批孖仔或者孖女啦，佢哋都係 identical 嘅 twins 同卵雙生嘅，即係佢哋嘅基因咧係一模一樣啊！我去探究一下啦，究竟啦有冇啲咩嘅因素咧係會影響到佢哋捲唔捲到你嘅呢個特徵嘅？下面呢幅圖咧就想顯示翻啦，呢一批嘅孖仔或者孖女啦，呢一批同卵雙生嘅人啦，佢哋同樣係捲到你啊，同樣係捲唔到你啊，同埋一個捲到你，另一個捲唔到你嘅百分比係幾多？咁當中咧有巧妙嘅，就係點解我哋要用同卵雙生嘅孖仔或者孖女呢？當中有咩嘅好處呢？咁首先咧你要去 recall 翻同卵雙生嘅定義係咩先？就係佢哋係由同一粒嘅精子、同一粒嘅卵子去受精作用啦，所產生嘅合子所變成嘅。即係話佢哋嘅基因咧係一模一樣啦。答案咧最基本你噏得出啦。同卵雙生嘅孖仔或者孖女咧，佢哋基因上係一模一樣嘅，即係話佢哋嘅基因型係一樣嘅。咁即係話啦，如果呢個特性係由遺傳嘅因素所去決定嘅話咧，佢哋應該係有同一個嘅表現型嘅。但係你會發現啦，有18% 嘅孖仔孖女咧，竟然係有唔同嘅捲脷嘅特性。有啲捲到，有啲捲唔到，唔係講緊咧一齊捲到或者一齊捲唔到。呢個就係 Part 2想問我哋嘅嘢啦。根據翻呢幅圖啦，就要揾翻啲證據去支持我哋呢兩個結論啦。第一個結論咧就係遺傳因素喺決定捲脷呢個特徵上面係扮演重要嘅角色。第二啦就係有其他因素影響咗捲脷呢個特徵。實質你要答啲咩呢？其實你就要知道翻喺 Part C 我哋嘅探究嘅目的係乜嘢啦。Part A、Part B 咧都清晰嘅。Part A 就想睇下啦，捲唔捲到你呢個基因咧，係咪真係由單一基因所控制啦？又或者邊個顯性邊個隱性啦？去到 Part B 啦，就幫我哋去揾得清啦。喂，其實當初咧錯地嘅，有冇其他因素係參與喺其中噶？Part C 就係話俾我哋聽呢一樣嘢啦。我哋就特登揾翻一啲原先基因型已經係一模一樣嘅人，再去睇下佢哋嘅表現型係咪真係一樣？點知就真係有啲唔一樣。咁呢個就係 Part C 想去探索翻有冇咩嘅因素，而呢個因素係除咗基因遺傳呢個因素之外呢，會唔會影響到捲唔捲脷呢個特徵呢？咁我哋發現原來得地喎。咁所以啦，要揾證據呢，其實同通識一模一樣嘅啫。資料回應題就係咁樣做嘅啫嘛，有幾分數據就講幾多分嘅說話。首先上面講嘅全因素喺決定捲脷呢個特徵呢，係扮演重要角色。咁我哋發現啦，因為有82% 嘅同卵雙生嘅孖仔孖女呢， 佢哋都係有同樣嘅表現型嘅。咁啊，同我哋嘅期望其實都相近㗎，同樣嘅基因型，有同樣嘅表現型，合理啦。但係啦，同樣一個實驗呢，我哋會得出另一個嘅觀察或者另一個結論，就係有其他因素牽涉喺其中啦。因為有18% 嘅同卵雙生嘅孖仔孖女呢，佢哋係表現到唔同嘅表現型嘅。咁究竟發生乜嘢事啊？正就係喺我哋書本都有講到嘅。同卵雙生大晒咩？基因型一樣大晒咩？環境因素都有機會去改變到一個人嘅表現型嘅。咁今次呢個例子就係講緊啦，大家都係孖仔孖女啦，同卵雙生嘅。咁啊，第一個人呢就食得好住得好嘅，咁啊肥屍大隻啦。另一個人啦，冇乜嘢食嘅，營養不良嘅，咁自不然就瘦猛猛啦。第一樣嘢呢，我想幫大家解一解讀嘅，就係咧，大家有機會誤會咗嘅，以為呢兩個結論呢係互相矛盾嘅，其實冇矛盾嘅，因為一個係講緊啦，基因遺傳嘅因素係重要嘅因素，我有冇講過佢係唯一嘅因素？嗱，如果我話佢係唯一嘅因素呢，第二句就變得有矛盾啫。但而家我只係講樓上係重要因素，咁即係有啲次要一啲啦。咁次要一啲嘅乜嘢啊？咪就係環境因素咯。好，跟住啦，去到 part D 啦。咁題目呢，要我哋去總結下啦。A、B、C 呢三個唔同嘅年代啊，捲唔捲到你呢個特徵嘅研究呢？呢個遺傳呢？我哋研究咗咁多嘢啦，整套嘅知識嘅發展呢，係能夠演示到邊一啲嘅科學理念呢？其實佢就係考緊我哋嘅科學本質。咁其實頭先嗰條題目呢，我約略都講咗噶啦。科學呢係一個不斷發問啊，係持續嘅過程嚟嘅。另一個科學本質呢，就係原來啦，科學探究係唔一定要喺實驗室嗰度做嘅。今次我哋要研究嘅嘢呢，係幾代人或者幾個家庭以上嘅資料嚟嘅。即係我哋唔會咧叫佢哋哇嗱嚟晒實驗室啦，跟住咧就交配啦，跟住生個仔俾我睇啦，唔需要嘅。我係透過可能問卷啦，我係透過觀察啦，記低翻啲數據啦，就已經得噶啦。佢父母親咧都係捲到嚟嘅，生到捲到嚟同埋捲唔到嚟嘅細路仔啦，佢哋數量係幾多咧？咁啊，原來啦，你一批咧捲唔到嚟嘅父母親咧，佢哋有超過30%嘅仔女咧都係捲到嚟嘅喎。又或者啦，我哋研究下呢啲孖仔孖女啦，咁究竟啦，佢哋係咪一齊捲到嚟，一齊捲唔到嚟，或者有啲捲到，有啲捲唔到呢？係睇都已經睇得到噶啦，你係唔需要韞佢哋喺實驗室嗰度做嘢嘅。特別我想講嘅咧，第三句就話啦，科學家呢，即使佢哋用同一款嘅數據，都係唔能夠得出同一個嘅結論嘅。點解呢句係喺呢個嘅發展入面顯示唔到呢？係因為啦，佢哋真係用緊唔同嘅數據啊嘛。A 有 A 嘅數據 ，B 有 B 嘅數據 ，C 有 C 嘅數據，咁佢得出唔同嘅結論，好正常啊，係咪？即使係 Part C，我哋拿住呢個 pie chart，同一款嘅數據啦，但係我哋有兩個結論，但係呢兩個結論係並存噶嘛，佢哋冇抵觸噶嘛，所以啦就唔會令到呢啲科學家話唔能夠去獲得同一款嘅結論。你望住呢個圖表，你就係有嗰兩個結論噶啦。基因遺傳嘅因素咧係主要因素，而亦都有其他因素咧係影響住捲唔捲到你呢個特徵。跟住到第二 part 咧，就要我哋去展述一下啦。喺整個 A、B、C 三個唔同年代啦，去研究下捲脷呢個特徵究竟點樣遺傳啊？點樣去展示得到？作為一個科學家係需要保持一個開放嘅態度呢？咁呢個題目呢，我哋又要睇下喺唔同年代呢，佢哋嘅探究嘅目的係乜嘢啦？1940年呢，我哋就想去研究一下究竟捲脷呢個能力係咪受到一個單一基因呢所控制？當其時嘅科學家呢，就得出一個結論：捲脷呢係顯性嘅特徵，而捲唔到脷呢，就係一個隱性嘅特徵。而去到65年呢，我哋就要驗證下啦，究竟捲唔到脷係咪真係一個隱性嘅表現型？點知呢，我哋就真係推翻咗1940年嘅結論。原來啦，捲唔到脷嘅父母親呢，都生到啲捲到脷嘅小朋友嘅。究竟發生咩事呢？去到71年啦，就開始諗下，除咗基因之外，有冇其他因素都會影響到捲脷呢個特性噶？就係原來呢，基因遺傳嘅因素係啦，其他嘅因素呢，都係有機會影響到一個人捲唔捲到脷嘅能力嘅。咁所以啦，原來咧科學家喺65年啦，同埋71年啦，其實佢哋都係不斷咁驗證緊佢哋前人嘅結果嘅。佢哋對於前人嘅研究咧，係保持住一個懷疑嘅態度。 而當中啦就去檢視啦，去挑戰啦當初嘅一啲理論啊，當初一啲知識。如果佢哋係冇一個開放嘅態度嘅話咧，永遠就係信佢啦，佢話係噶啦。喂 Alfred 啊係一個好出名嘅科學家嚟噶，你唔係質疑佢啊嘛，好唔尊重咯。OK， 我哋就透過實驗去研究1、2、3，我哋就係透過數據嘅分析，我哋就係透過科學探究嘅形式去收集唔同嘅數據，去睇下究竟當初嘅結論正確抑或唔正確。咁話唔定係正確噶嘛，係咪？咁冇人話去檢視你嘅結果就係唔尊重你咩？唔會噶嘛。喺科學探究好公平嘅啫，數據就說話噶啦。呢個年代喺呢個科技嘅層面底下，你獲得呢一個嘅結論，冇人反駁到你呢，我哋就信住先。去到下一個年代，有另一批嘅科技，有另一啲嘅方法嘅話呢，我哋獲得一個新嘅結論呢，我哋就能夠修正翻過往所留落嚟嘅知識同埋理論啦。好，咁嚟到一點出發啦。今次嘅題目呢，就講捲脷嘅，咁啊考我哋兩樣嘢啦，基礎遺傳學啦，同埋呢個同卵相生嘅。咁啊基礎遺傳學啦，佢可以考我哋啦分離定律啦，咁啊即係曼德爾第一定律啦。咁啊因為啦今次我哋淨係睇單一基因啫，咁當然啦到頭來我哋發現呢，佢未必完全係單一基因控制住嘅，可能仲有環境因素啦。但係點樣都好啦，俾個族譜你啦，我睇下 Peter 啦，睇下 Mary 啦，一個捲脷，一個捲唔到脷嘅。究竟啦佢哋個仔女捲到脷嘅機會率係幾多呢？叫你話個遺傳圖都得嘅，咁啊分得清個基因型啦，同埋表現型啦。咁啊去到啦同卵相生呢，咁啊排卵啦同埋受精啦。咁呢個係人類繁殖嗰個 topic 啦。咁如果翻去 basic genetics 嘅話呢，就起碼我哋知得到啦。同卵相生嘅孖仔孖女咧，佢哋基因上係一模一樣嘅，所以佢哋性別咧亦都必然一樣嘅。而另一樣嘢咧要考我哋嘅啦，就結合翻基礎遺傳學嘅概念，就係變異啦。咁點解唔同嘅生物啦，即使佢哋係同一款嘅物種，大家都係人類啦，但係點解我哋嘅高矮肥瘦啊、皮膚顏色啊、頭髮顏色啊可以唔同咧？咁呢個就係變異啦。咁題目可能問下我哋啦，點解變異會出現呢？咁其中一個原因啦，就係遺傳基因嘅問題啦。大家嘅父母親都唔同啊，大家基因嘅組成都唔同啦，又或者啦，受到嘅環境影響咧都唔同嘅。咁另一款啦，再進深啲嘅咧，就係問下你基因遺傳點解會出現啦？好常問噶啦，基因嘅變異係嚟自於，例如啦，減數分裂嘅時候啦，染色體嘅獨立分配啦，又或者啦，製造咗個配子之後啦，精子同埋卵子之間一個隨機嘅受精作用啦，又或者基因突變啦，如此種種啦。咁呢個題目咧都有考到我哋咧有關於科學本質嘅。點樣可以成為一個科學家呢？原來要 open mind， 或者從個數據當中點樣獲得結論呢？點樣利用數據呢？咁呢個呢，大家通識科呢都有學過嘅，又或者而家公民科啦都輕輕嗨到一啲啦。咁過往啦你見得到啦，13啊、15啊、16啊，其實都有咧遺傳學嘅題目嘅。今年2017年都唔例外嘅，所以基因遺傳呢，真係一個熱門課題嚟嘅。咁多位。17, question 10 is the question of basic genetics. And the topic we are studying is the ability to roll the tongue. So in 1940, a scientist, he hypothesized that the ability to roll one's tongue is determined by a single gene. Therefore, he collected different data to prove that, to investigate whether he is correct or not. So for the case 1, so the characteristic of the parent, both of them, they are tongue rolling, and then we can do the statistic, how many of the offspring they can roll their tongue or they cannot roll their tongue. And for the other case, one of the parents, they are non-tongue rolling, and then we also collect the data to see the statistic, how many of the offspring they can roll their tongue or they cannot roll their tongue. So for part 1, does the trait of the tongue rolling ability show continuous or discontinuous variation? Explain your answer. So in this question is checking us the concept to distinguish the continuous variation from the discontinuous variation. And then we can see the basic definition. We can see that for the discontinuous variation, we can see the distinctive phenotype without intermediate. So in this case, for the trait, there are only two types, two categories. One is tongue rolling and the other one is non-tongue rolling. Tongue rolling ability trait is the discontinuous trait because they are distinctive categories with no intermediate categories. And for part 2, the scientists concluded that tongue rolling is the dominant phenotype while non-tongue rolling is the recessive phenotype. So with reference to the above table, that means the data he collected explain how he arrived at this conclusion. For this question, we need to apply the critical skills out of nothing triangle and the 4 steps genetics scaffolding. So for the out of nothing triangle, we need to choose which case we should use to do the deduction, and then to conclude that tongue-rolling is the dominant phenotype and the non-tongue-rolling is the recessive phenotype, because it meets the requirement of out of nothing. Tongue-rolling times tongue-rolling, and then there are non-tongue-rolling offspring. So you can see that out of nothing. And then how can we present our answer? So we use the 4 steps scaffolding. So first step, we identify the parent's phenotype and deduce the possible genotype. Step 2, we need to identify the particular offspring's phenotype and deduce the possible genotype. And for step 3, we need to identify who is or probably the heterozygous individual. In this case, we can see that the parents in case 1, they are heterozygous. And for step 4, we can determine the dominant and the recessive allele. First of all, we need to mention that the case 1, that's the data we are going to use. And then we take a look at the offspring. Non-tongue-rolling offspring appear even when both parents were tongue-roller. It means that they must inherit at least one allele for non-tongue-rolling from at least one of the roller parents. That's the reason why we can say that the parents in case 1, they are heterozygous, because for themselves, their phenotype is tongue-rolling. At least one of them, they have at least one non-tongue-rolling allele. So it shows that the allele for non-tongue-rolling was masked in the parents. And then we can use the idea of the 4 steps. For the recessive allele, it will be masked in the heterozygous condition. Therefore, tongue-rolling ability, it is the dominant phenotype, and the non-tongue-rolling ability is the recessive phenotype. You may ask that, Mr. Leung, why there are only 2 marks in this question? For the normal 4 steps genetics scaffolding question, there should be 4 marks or even 5 marks. Because step 3 and 4, we don't need to mention it. They are already given by the question. After the investigation in part A, 1940, we have a lot of data to help us to conclude that the tongue-rolling ability is dominant characteristic, and for the non-tongue-rolling ability is the recessive characteristic. So for part B, we have another research. In 1965, the offspring of a group of non-tongue-rolling parents were studied, and it was found that more than 30% of the offspring were tongue-roller. So does this finding support the conclusion we draw in part A2? And explain your answer. For the whole thinking logic of part B, we need to know that according to part A2, non-tongue-rolling is the recessive phenotype, while the tongue-rolling is the dominant phenotype. So if we use the symbol to represent them, so the capital letter T, it is the tongue-rolling allele. And for the small letter t, it is the non-tongue-rolling allele. So according to part B, a group of non-tongue-rolling parents were studied. Both of them, they must be the homozygous recessive, small t. So based on this logic, and then you can use 10 seconds to finish the genetic diagram. So you can find that if both of the parents, they are non-tongue-rolling, and as the conclusion mentioned in the part A2, both of them, they are the homozygous recessive. So all the offspring, they must be non-tongue-roller. However, in 1965 study, we find that more than 30% of offspring, they were tongue-roller. Therefore, the findings, it doesn't support the conclusion, because all the offspring of the non-tongue-rolling parents be non-tongue-roller. However, in fact, they are more than 30% of offspring, they were tongue-roller. In part B, it shows us the nature of science. Scientific knowledge is tentative and subject to change. So in part A, we concluded that tongue-rolling is the dominant phenotype and the non-tongue-rolling is the recessive phenotype. However, in part B, the data make us doubt what we believed before. And then that's why we need to keep asking questions. What's going on inside? It's also a way for us to make the science advance, to improve. So after part A, part B investigation, it seems like that we do not have a clear understanding of the inheritance of the tongue-rolling ability. Therefore, in 1971, we have another study. on identical twins in order to explore the factors influencing the tongue-rolling trait. Then we can see the result in this pie chart. 71% of the identical twins, they are both tongue-rolling. 11% of the identical twins, they are both non-tongue-rolling. And 18% of them, one was tongue-rolling and the other was non-tongue-rolling. So for part A, it asks us that why do we use the identical twins as the subject for the study? Any advantage to using the identical twins? So the concept checking, we need to recall the definition of the identical twins. They are formed by one ovum fertilized by one sperm, form one single zygote. For the identical twins, they have the same genetic makeup, same genetic composition. And what if the trait is controlled by the genetic factors? That means they have the same genotype, so we expect that they should have the same phenotype. However, you can see that 80% of them, they show different phenotype. So that's the problem. What's going on actually? So we move on to part 2. With reference to the above chart, we complete the table with the data that support the conclusion. So what are the two conclusions? First of all, genetic factors play a significant role in the determination of the tongue-rolling trait. And the second conclusion, there are other factors influencing the tongue-rolling trait. So what is the thinking logic of this question? We need to recall the aims of the investigation in part A and part B and also part C. So in part A, we concluded that the tongue-rolling ability is dominant and the non-tongue-rolling ability is recessive. But in part B, it seems like that the conclusion is not that correct in 1940. Of a group of non-tongue-rolling parents, they can give birth to tongue-rolling offspring. So it makes us to think about that, is the genetic factor the only factor to control the tongue-rolling ability? If not, so what are the other factors? So that's why we have part C. So in part C, we use the identical twins who are genetically identical to explore any factor which is other than genetic factor, which also influence the tongue-rolling trait. And we really find that some of the identical twins, one was tongue-rolling and the other was non-tongue-rolling. So how can we draw the conclusion? It's just like that in the liberal study data response question. Tell what you observe, because you can use the data directly. How can we support the first conclusion? There are 82% of the identical twins show the same phenotype. So what about the second conclusion? There are some other factor influencing the tongue-rolling trait, because there are 18% of the identical twins, they show different phenotype. So what I would like to remind you is that don't think that these two conclusions, they are contradictory. They are not contradictory. I'm not saying that the genetic factors, they are one and only one factor to determine the tongue-rolling trait. If I say so, these two conclusions, they will be contradictory. But now I'm just saying that the genetic factor, they play a significant role. So what about other factors? Other factors, it also count. For example, the environmental factor. So what we learned from the textbook is that for this pair of identical twins, one is brought up in a well-nurtured so one of them, they have good nutrition, good environment, so he can grow faster, rapidly. But for the other one, poor nutrition. So that's why he will become a bit skinny, a bit short. For part D, in above case regarding the development of the knowledge about the inheritance of the tongue-rolling trait, which of the following idea about the science is demonstrated? Means we need to summarize all the investigation in part A, B, C. What are we doing? For example, we would like to prove that the ability of tongue-rolling is determined by a single gene. Is it the tongue-rolling ability is the dominant phenotype, or any other factor will also affect the tongue-rolling trait? So we need to recall the nature of science. Science is a process of ongoing inquiries. That's something I have already mentioned in previous slide. Second nature of science is that scientific investigation may not require doing experiment in the laboratory. We collect the data from the parents, both of them they are tongue rolling, and then count the number for the statistic, how many of the offspring they can roll their tongue, or we collect the data of the offspring of a group of a non-tongue rolling parents. So let's see how many of them they are tongue roller. Or we collect the data from the identical twins to let's see are both of them tongue rolling or non-tongue rolling, or one was tongue rolling or the other was non-tongue rolling. So we just do it in the office is okay. We send out the questionnaire and then we collect the data and then it will be fine. And I would like to talk about the third one. Scientists may not arrive at the same conclusion about the same set of data. It cannot be shown in this example because they are using different sets of data. In 1940, we have a set of data. 1965, we have another data. In 1971, we have another data. So we use different data, or we have different data to arrive at different conclusion. It's normal. Even in part C, we use the same data, and then we have two conclusions. But that's really what we observe. 82% of the identical twins, they show the same phenotype, and then it shows that the genetic factor, they may play a significant role on the inheritance of the tongue rolling trait. And then the second conclusion, some other factor will also influence the tongue rolling trait. It's also supported by the data. And then for part B, we need to elaborate on how the development of the knowledge about the inheritance of the tongue-rolling trait can be used to demonstrate that scientists have to be open-minded. Or we need to recall the aim of the investigations. For 1940, we need to prove that the tongue-rolling ability is determined by a single gene, and at that moment, we concluded that tongue-rolling trait is dominant and non-tongue-rolling trait is recessive. However, in 1965, we would like to prove that non-tongue rolling ability, is it really recessive phenotype? But we can see from the data, the non-tongue rolling parents, they can give birth for more than 30% of offspring, they were tongue roller. So somehow we disprove the deduction of the dominant and recessive phenotype in 1940. And then in 1971, we would like to study if there are any factor influencing the tongue rolling trait, because in 1940, 1965, it seems like that the genetic factor, not the one and only one factor. So after the investigation, we conclude that the genetic factor and other factors, they will exert the effect on the tongue rolling ability. So we can see that scientists who conduct the tongue rolling experiment or research in 1965 or in 1971, they need to be skeptical and look for evidence from different sources, different perspectives. Therefore, they review or challenge the current ideas, the knowledge of the theories. So you can see that what if the scientists, they are not that open-minded, and if they think that no, Alfred, he is a very famous scientist, he is the authority or he is the expert to investigate the inheritance of the tongue rolling ability, we cannot challenge him. Therefore, there will be no 1965 investigation and 1971 investigation. Or at this moment, we still believe that tongue rolling is dominant phenotype and non-tongue rolling trait is the recessive phenotype. Let's talk about the curriculum mapping. This question starts from the concept of tongue rolling and it checks the concept about the basic genetics and the identical twins. So for the basic genetics, it may ask you about the law of segregation because there is talking about one gene inheritance. And apart from the genetic diagram, it can also ask you about the pedigree. You study the whole family, or Peter is tongue rolling and Mary is non-tongue rolling, something like this. And then to study their genotype and phenotype. And for the identical twins, so for the ovulation, fertilization, that's the concept of the human reproduction. From the concept of basic genetics, we know that identical twins, they are genetically identical. Therefore, they must have the same sex. And then we can recall the concept of the variation and then combine it for the scientific investigation in this question. So for the variation, any possible question variation, it can ask you that why are there variation in the organism? So it could be the result of the hereditary heredity and or the effect of the environment. Or next time it can ask you that how are genetic variation brought about? For example, the independent assortment of the chromosome at meiosis, crossing over at meiosis, random fertilization and mutation, something like this. And in this question, it also asks us about the nature of science. So it helps us to expand the idea how to be a scientist. So you can see that in 213, 15, and 16, there are lots of questions about the genetics. So you can see that it is really a hot topic.